Alright, so in this video we're going to go over protein structure, primary through quaternary, and we're going to do so with the help of pipe cleaners. So, what are pipe cleaners made of? They're made of fuzz and wire, right? So, they're made of fuzz, and they're joined by wire. How would you separate a pipe cleaner? One could imagine with scissors, possibly, right? I don't know if anybody's got a pocket knife. That could work too. And then, will the pipe cleaner function the same after we cut it up into pieces? No. A pipe cleaner is long, and it's supposed to be able to bend into all these different shapes. But if you cut it into pieces, it's not going to be able to do its job. Right, so how is this like a polypeptide? Well, a polypeptide is made up of monomer subunits known as amino acids. And they're joined by the bonds that form between amino acids known as peptide bonds. How would you separate the amino acids that are joined together by peptide bonds? Enzymes. You would need protein enzymes to do so. An example of this is, I believe, pepsin in the stomach, which functions at a super low pH. And will the polypeptide be the same after being cut up into pieces? No. As same thing with the pipe cleaner. If it's cut into pieces, it can't fold and do its function. So now we're going to go into the folding aspects. So with secondary structure, we're going to form alpha helices and beta sheets. So with this red pipe cleaner, I'm going to form an alpha helix. I'm going to twirl it around my finger like that. And we have approximately an alpha helix, so we'll set that over there. The blue pipe cleaner will make a beta pleated sheet. This has got lots of fun amino acids in it that allow it to curve back and forth. Alright, so that's a beta pleated sheet. And then on one of these yellow pipe cleaners, we're going to try and do both. We're going to make a little bit of an alpha helix and then just a bit of a beta pleated sheet to show that these can happen in the same polypeptide. Just depends on what amino acids are in there, right? So secondary structure depends on the primary structure, the sequence of amino acids. So secondary structure is stabilized by what bonds? These would be hydrogen bonds that are formed between the polar backbone. So if you remember from the amino acid video, whenever we form the peptide bond, there are the leftover remnants of the amino group and the carboxyl group in the backbone, and those are what hydrogen bond to form alpha helices and beta pleated sheets. So, how would we straighten out the amino acid after, or how would we straighten out the pipe cleaner after forming one of these? Well, I could just use the force of my hand, right? That's easy enough. Just use force. You don't need an iron or anything. And how would you straighten out a polypeptide? Well, that's going to be a special process known as denaturation, which we'll cover soon. Alright, so hydrogen bonds are not exclusive to polypeptides and proteins. They're also found in carbohydrates. My favorite example would be cellulose, the most abundant carb on the planet, and nucleic acids, right? So hydrogen bonding is going to happen between the different base pairs. And I'm sure you can probably find a couple of examples of hydrogen bonding in lipids, but it wouldn't be as present because lipids are a bit more hydrophobic and hydrogen bonding happens between hydrophilic portions of molecules. Alright, so tertiary structure is stabilized by bonds that occur between the R groups, we call it in the amino acid video, of non-neighboring amino acids. So that's the big confusion that students tend to have, is the difference between secondary and tertiary structure. A lot of students will say that secondary structure is just alpha helices and beta sheets. But what we're looking for is that secondary structure is between the backbone and tertiary structure is between the R groups. Because tertiary structure also includes hydrogen bonds. So you can't just say hydrogen bonding is secondary structure because it's also tertiary structure. But it's between the backbone in secondary structure. And tertiary structure is between the R groups. Other types of bonds that are also present in tertiary structure would be ionic bonds, where an electron is donated to another amino acid, R group. We have disulfide bridges, which are specifically formed between the cysteines of amino acids. And we also have hydrophobic interactions, which aren't necessarily bonds, but more so parts of the protein that don't want to interact with water will instead just interact with each other. The oily amino acids will interact with other oily amino acids. Right. Alright, so how would you straighten a pipe cleaner in its tertiary structure? We must show tertiary structure on these different bits. So normally we would use a purple pipe cleaner. I do not have any purple pipe cleaners on me, so I'm going to use just a bit of duct tape. I would say that the duct tape probably most closely mimics the disulfide bridge because it's kind of the strongest, because it's a covalent bond. Right, so there's our tertiary structure. We'll just allude that like such. There's our little disulfide bridge. Could be any of them, but I'm going to say it's a disulfide bridge because I'm using duct tape. And duct tape's a bit, actually Gorilla Tape. Okay, so how would you straighten the pipe cleaner? You have to pull it apart. Once again, just use force. A bit more force this time, though. With a polypeptide, you're once again going to be using denaturation. So what is denaturation? Well, denaturation is the process of breaking down the secondary and tertiary structure of proteins. And we know denaturation very well. This can be done through heat, which we know is cooking. You can cook an egg. It goes from clear to white. That's the different proteins denaturing and getting all scrambled up. You can use acid and marinating. This is how you effectively cook fish in ceviche. And then you can also use force and whipping. This would be like making a meringue. You whip the egg whites into the big fluffy meringue that you put on top of pies and stuff. So, compared to breaking down the primary structure, what would be the breaking down of that? We've covered this before many times. Hydrolysis. We covered that in the last video. You break down the primary structure, the sequence of amino acids, through hydrolysis. You add some water. To do this, you can't just simply add heat or acid or force. You need protein enzymes. So this process is sort of regulated by the cell. Different proteins get ubiquitinated and get thrown into a chemical trash compactor because they need enzymes to do this. Alright, so finally we're going to end with quaternary structure, which is multiple subunits coming together. So let's say that this right here is going to be a hydrophobic domain. So whenever we bring all these subunits together, we kind of want to shield this, because if this is going to be hydrophobic, the outside of the protein is going to be in water. The hydrophilic part should be facing outside, the hydrophobic part should be inside. So we'll put our subunits together and sort of shield that portion. It's going to be on the inside of our protein. We want to make sure that it does not have to see scary water. Alright. So this could be our final protein, right? We have hydrophobic on the inside and hydrophilic on the outside. Now if you submerge this protein in oil, you can just imagine that it would flip right inside out. All the hydrophobic parts would be on the outside, all the hydrophilic parts would be on the inside. But that's an oil. We live in water, so it's different. And so finally, is quaternary structure essential? No. There are many proteins that exist just as a single polypeptide. You don't need multiple polypeptides to come together. However, there are many important proteins, such as hemoglobin, that exist as dimers, tetramers, trimers, whatnot. Hemoglobin is a tetramer. It has 4 different domains. So quaternary structure could be essential for some proteins, but it's not essential all the time. So if you have pipe cleaners at home, go ahead and try and make one of these on your own. It'll be kind of fun. You can hang it up on your wall. They're super cute.